In the hushed annals of scientific history, certain moments stand as stark reminders of humanity's relentless pursuit of knowledge, often teetering on the precipice of the unknown. What happens when ambition collides with ethics, when the very definition of life is challenged? This is the chilling, yet profoundly informative story of an experiment that dared to blur the lines between species, igniting a global firestorm and forever altering the landscape of bioethics. The year was 2003. In the bustling metropolis of Shanghai, within the seemingly innocuous walls of the Shanghai Second Medical University, a clandestine endeavor was underway. Dr. Hui Zhensheng and his team embarked on a project that would send shockwaves across the scientific community and beyond. Their goal was audacious, their methods controversial. They sought to create something unprecedented, a human-animal hybrid embryo. This wasn't the stuff of science fiction, it was a tangible, microscopic reality. The world would soon grapple with the implications of what some would controversially dub the Bunny Man experiment. To comprehend the profound implications of this experiment, one must first grasp the underlying scientific methodology. The technique employed was somatic cell nuclear transfer, or SCNT. This is the very same process that famously brought Dolly the sheep into existence, marking a pivotal moment in cloning history. In SCNT, the nucleus, which contains the genetic material, is carefully removed from an unfertilized egg cell. This enucleated egg then receives the nucleus from a somatic cell, any cell from the body other than a sperm or egg cell of the organism to be cloned. The reconstructed cell is then stimulated to divide, ideally forming an embryo. In Shanghai, the team utilized rabbit egg cells, chosen for their availability and ease of manipulation. From these, the nuclei were meticulously extracted. Into these enucleated rabbit eggs, human somatic cell nuclei were inserted. These human cells came from two distinct sources, a 2-year-old boy and a patient suffering from psoriasis. The resulting cellular constructs were then stimulated to develop. The objective was to observe their progression to the blastocyst stage. This is a microscopic cluster of cells, typically formed within 5 to 9 days after fertilization, containing an inner cell mass that can give rise to embryonic stem cells. It is crucial to emphasize that no fully formed creature, no bunny man, ever materialized. This was an experiment conducted at the cellular level, a fusion of human and animal genetic material, in its earliest, most fundamental stages. Why undertake such a controversial experiment? The scientists were not driven by a desire to create monstrous chimeras. Their motivations were rooted in a pressing challenge within the burgeoning field of stem cell research. The ultimate goal was therapeutic cloning. Therapeutic cloning aimed to generate patient-specific embryonic stem cells. These cells, with their remarkable ability to differentiate into any cell type in the body, held immense promise. They could potentially be used to grow new tissues or organs, offering revolutionary treatments for debilitating diseases like Parkinson's, Alzheimer's, diabetes, and spinal cord injuries. The vision was to create perfectly matched cells, eliminating the risk of immune rejection. However, a significant hurdle existed: the scarcity of human egg cells. Obtaining human eggs for research purposes is ethically complex and logistically challenging. It requires invasive procedures for donors and raises concerns about exploitation. The Shanghai team sought an alternative. The rabbit egg, with its readily available cytoplasm, was seen as a potential solution. The cytoplasm of an egg cell contains vital factors that can reprogram a somatic cell nucleus back to an embryonic, pluripotent state. The hypothesis was that the rabbit egg's cytoplasmic environment could effectively reboot the human nucleus, allowing it to behave like an embryonic nucleus and generate stem cells. This innovative approach aimed to circumvent the ethical and practical limitations associated with human egg donation, accelerating the pace of medical discovery. The revelation of the Shanghai experiment ignited an immediate and ferocious ethical firestorm across the globe. News headlines screamed, ethicists debated, and religious leaders condemned. The very notion of blending human and animal genetic material struck a deep chord of unease, challenging fundamental perceptions of identity and species boundaries. A central point of contention revolved around the moral status of the hybrid embryo. When does life truly begin? Does a microscopic entity, containing both human and animal DNA, deserve the same protections as a human embryo? These questions, already complex in the context of human cloning, became exponentially more fraught with the introduction of animal components. The concept of human dignity was invoked, with many arguing that such experiments inherently devalued human life. The blurring the lines of species argument gained significant traction. Critics voiced profound concerns about the potential for creating chimeras that could erode the distinctiveness of humanity. This wasn't just about scientific curiosity. It touched upon deeply held philosophical and theological beliefs about the natural order. The fear of playing God resonated widely, particularly within religious communities, who saw the experiment as an affront to divine creation. Perhaps the most pervasive fear was that of the slippery slope. If human-animal hybrid embryos were permitted, what would be next? Would scientists eventually attempt to create more advanced chimeras, perhaps even sentient beings with mixed characteristics? This speculative but potent concern fueled public anxiety and calls for strict regulatory oversight. There was also a theoretical, though unproven risk of novel hybrid diseases emerging from such interspecies genetic mixing, adding another layer of apprehension. The controversy manifested in public protests, heated parliamentary debates, and a flurry of legislative proposals aimed at curbing such research. The immediate consequence of the Shanghai experiment was a swift and decisive global reaction. Governments and regulatory bodies, already grappling with the ethics of human cloning, moved to establish clearer boundaries. Countries like the United Kingdom, Canada, and Australia, already at the forefront of bioethical legislation, quickly enacted or reinforced laws specifically limiting the gestation period of human-animal hybrids. These regulations typically permitted such constructs only for a few days, strictly prohibiting their development beyond the blastocyst stage and certainly forbidding implantation into a womb. The Bunnyman experiment, despite its initial scientific ambition, was not widely replicated. This was due to a confluence of factors, both ethical and scientific. The intense public outcry and the subsequent legislative restrictions created a formidable ethical barrier. Scientists, keenly aware of public perception and funding implications, largely shied away from pursuing similar avenues. Scientifically, the experiment also revealed significant hurdles. While the rabbit egg cytoplasm could initiate reprogramming, the resulting hybrid cells often exhibited genetic instability. There were also concerns about mitochondrial mismatch. Mitochondria, the powerhouses of the cell, contain their own small amount of DNA. When a human nucleus is placed into an animal egg, the human nuclear DNA interacts with the animal mitochondrial DNA, which can lead to inefficiencies or even cellular dysfunction. These technical challenges, combined with the ethical quagmire, made the approach less appealing for long-term research. The Shanghai experiment thus became a cautionary tale, a stark reminder that scientific advancement must always be tempered by profound ethical consideration. Paradoxically, the controversy surrounding the Shanghai experiment, and indeed the broader debate on embryonic stem cell research, inadvertently spurred a monumental breakthrough. The intense pressure to find ethically less contentious alternatives galvanized researchers worldwide. This urgency paved the way for a discovery that would revolutionize regenerative medicine: induced pluripotent stem cells, or iPSCs. In 2006, just 3 years after the Shanghai experiment, Japanese scientist Shinya Yamanaka achieved what many thought impossible. He demonstrated that adult human somatic cells, such as skin cells, could be reprogrammed back into an embryonic-like, pluripotent state. This was accomplished by introducing just a few specific genes into the adult cells. These reprogrammed cells, now known as iPSCs, possess the same remarkable ability to differentiate into various cell types as embryonic stem cells, but without the need for embryos or animal eggs. The development of iPSCs offered a truly transformative solution. It provided a way to generate patient-specific stem cells without the ethical dilemmas associated with human embryos, or the interspecies concerns of hybrid constructs. This breakthrough earned Yamanaka a Nobel Prize and fundamentally shifted the trajectory of stem cell research. It offered a more ethical, practical, and widely accepted pathway for developing regenerative therapies, effectively rendering the human-animal hybrid approach largely obsolete for therapeutic purposes. The Bunnyman experiment, while controversial, inadvertently accelerated the search for alternative solutions, ultimately leading to a more promising and ethically sound future for regenerative medicine. The Shanghai experiment, a fleeting cellular entity, was never a monster in the literal sense. Yet, its symbolic power was immense. It served as a groundbreaking, albeit ethically fraught, proof of concept, demonstrating the plasticity of genetic material and the potential for interspecies cellular manipulation. More importantly, it became a pivotal moment in the ongoing global dialogue about bioethics. This experiment, alongside other early stem cell research, forced humanity to confront profound questions about the boundaries of life, the definition of species, and the moral responsibilities of scientific inquiry. It highlighted the critical need for robust ethical frameworks and public engagement in guiding cutting-edge research. Its legacy is not just about what was created, but about the intense ethical discussions it provoked and the subsequent drive it provided for alternative, more acceptable scientific pathways, culminating in the advent of iPSCs. As we stand on the cusp of unprecedented biotechnological advancements, the lessons from Shanghai remain profoundly relevant. The future of medicine promises incredible breakthroughs, from gene editing to synthetic biology. But with every new frontier, the ethical considerations become more complex, more urgent. We must continue to ask, just because we can, does it mean we should? The story of the Bunnyman experiment is a powerful reminder that true progress lies not just in scientific innovation, but in responsible innovation, guided by wisdom, foresight, and a deep respect for life itself. What new ethical dilemmas will tomorrow's science unveil? How will we, as a society, navigate the uncharted waters of future biotechnologies? The conversation is far from over. Join the vital discussion. Share your thoughts on the ethical boundaries of scientific research in the comments below. Subscribe to our channel for more in-depth explorations of science, history, and the profound questions that shape our future. Your engagement fuels this crucial dialogue.